Okay, so bioinformatics, as I told you, so it's the marriage between, let's say, computer science and biology. Okay, so there are two aspects. So you know there's a bioinformatics subject offered for biology students as well. So this course differs from that course mainly from, we are looking at how can we develop the tools, how can we improve the tools for this biological data analysis, rather than how can we apply our computational methods rather than applying them, okay? Maybe the zoologists or these biology people, what they do is they use the tools developed by yourselves and they analyze data, okay? We can do both. We can, we have to, our main purpose of this course is to learn what algorithms, tools and techniques can be applied. If so, how can we improve, okay? How can we develop and how can we improve these computational techniques to apply on this biology data. So there are different definitions for bioinformatics. So most common definition is the what I have given you, it is the marriage of computer science and biology. So, so one can say it's the information technology applied to management and analysis of biological data. And you know, this biological data is vast. Why do we say so? So if you look at one person to the other, like in your batch, there are 7 people. If you look at Sean and Mevan, 2 boys, I think. So if you look at those 2 people, they are different, isn't it? Although they can eat, they can see, they can sleep, they can do all the physical work, common physical things. However, they look different. They can do both common things. They can study, I mean, to some extent, to some different levels. However, they have some common features. However, they are different from each other, isn't it? So if you look at a human and a cat, they're totally different. Okay. So because of these reasons, these characteristics are coming from the genes which are expressed in this people's and cats or the 2 different persons' body. So you can see these differences coming from each and every person in an organism like humans, like Mevan and who is that, Dylan, I can't remember the name, are different each other because they, in their body, these expressed genes are different from each other. So these variations of gene expression differences, and hence the different proteins are expressed. So there's a wide variety of proteins expressed in different people's body, and different these organisms like humans and cats, they also differ from these gene differences, okay? Genome differences. So a lot of new terms maybe you may think, like genes, genomes, proteins, what are they? Okay, so in this today's lesson, the main target is to discuss about the biological background required throughout this lecture series. So we'll give the basics, we'll learn the basics today. So don't be afraid, we are not learning biology as a biologist, okay? We only learn which is required to understand these techniques we are going to apply, okay? So that is the limit. So, I mean, if you are interested, then your main target is to be a scientist one day, maybe it's not, maybe sometimes in, I don't know, Sri Lanka, whether we have such good infrastructure for that. In other country, or if you are going for a PhD, you have dreams of higher studies. So it's really important to look into the different aspects, but as a bioinformatician, you are not going to learn biology as a biology student. Okay, you are just studying what you want. So when you are selecting your research also, I mean, I'm not specifically talking about bioinformatics research. So you will study a particular domain. Okay, so if you are going to apply these computational methods to classify newspaper items, then you will study about that domain. What are the keywords? So likewise in this bioinformatics also, there are certain keywords we need to identify in deep. Okay, so that's what we are doing today. The basics which we require would be discussed, and through a group activity, we'll, I mean, just this would not be a traditional lecture, this bioinformatics. I always wanted to encourage students to enroll more. So if you ask your seniors, there would be they would say that there were many practical activities organized. Online also I did them, because this group in thing is available in these breakout rooms. So they did the presentations, sharing their screens. So don't worry that we are doing, maybe this activity would be less in online. It would not be less, it would be more. Okay, so don't worry about that. There would be no any differences whether you are doing physically or online in this course. Let's start. So as I told you, this bioinformatics is the marriage between computer science and the biology, and there are different definitions available. And the main goal is analyze and interpret these various types of data, gene-related data, protein-related data, mRNA-related data, to understand biological problems. Okay, we are going to apply the computational algorithms, tools and techniques like neural networks, mainly algorithms, neural networks, and SOMs, self-organizing maps. Likewise, we are trying to apply few, and we are trying to look at how these can be developed, how these tools can be developed. Okay, so that's the main goal of this course. So as I told you, bioinformatics is a multidisciplinary subject. Okay, so so when you be a bioinformatician, so you need to know, and it means not knowing actually. When you are doing a PhD on bioinformatics, you would have to learn some chemistry, some physics, some statistics, some maths, and some biology. Okay, so as a computer scientist, you have that knowledge, and also you have to learn. Learning the new technologies is never ending. We also do courses. We also update our knowledge. Okay, so bioinformatics. Why do you think we need such a lot of areas to understand to develop some tools to understand biological data? Do you have idea? Why do we need chemistry and physics? Especially the other things you can guess, isn't it? Statistics, maths. It's mainly because there's a lot of data available. Biology, computer science are must. Why chemistry and physics? Do you have an idea? Can you tell me some idea? Ashpa, what do you think? Why do you need chemistry and physics? Mahoga?
It has the basic concept of the system.
Why do we need chemistry? Yeah, they have basic concepts. Yeah. How do we relate this thing with biology, especially? So you get fever, isn't it?
Yeah.
You get fever, and your parents may have high blood pressure, isn't it? And you may have some disease related with chemical components. So this pressure and temperature related things require physics related related principles and chemistry related principles to when you go into your body, it's a collection of chemicals. Okay, so today you'll understand that. So that's why I told you this subject is really interesting. So once you know this, you have to know as a human these basic concepts. How are we built up and what is actually happening in our body, and what are the techniques available to do these treatments, novel treatments. So those, when you know this, at least when your child asks, Papa, I mean, how am I built up of? You should have answer, isn't it? So this is not only a subject in this field, also this would open up your, I mean, vast amount of knowledge for your life. Very important, useful information, okay? So I need your active participation when I ask a question. Don't be shy. Everyone would make mistakes in their life, okay? So it's your friends and you are a family now. You have to, go through. Don't be afraid. You have to know each other. Answering the question, it should not be a very big task for you, okay? You have been into the industry now. You know how communication is important, isn't it? So let this be another opportunity for you to improve your communication skills as well. So at the end, in your projects also, what we see is actually you have improved a lot. When you first do a proposal, it's not you actually. At the end, we can't actually imagine how you improved. Okay, so such an improvement is required from you as well. In your seniors, we saw that. The first start, they are very shy. They are lack of knowledge. But at the end, they go through very well and they go for international journals as well with their work. So there's much to do. So be prepared and take the opportunities. Okay, recently there was a hackathon. I think you got some information about that. So participate in them as a whole group. You can work together and go for them and work together, and you can achieve very good results, because you are the very best people in this batch. You are the gist of this batch, as we say. So you need to perform very well, and it's expected from the department as well. Okay. So these are the main topics covered in this subject area. Introduction to Bioinformatics and Molecular Biology. Today we'll be doing that. So all the required biological concepts, basic concepts will be discussed today. And then next week we'll be discussing about archives and biological information retrieval. That would go around 2 weeks because you need to know ways of retrieving biological data. Although we say that these people has different genes, how can we get these genes for our analysis? So that is what we are discussing here. Okay, both of these topics, archives and biological information retrieval and database search. Then we'll be applying this, applying the tools available. We'll applying these computational methods on this data we discuss in these 2 lectures. So after that, the application 1 is alignment. So we are trying to align them. What is alignment? We are trying to match one gene sequence with other sequence and try to find out some similar regions. Okay, so that is what done in alignments. And in phylogenetics, what we are looking at, we are trying to look at how we are related with each other. So when you look at the genes, there are more than 98% similarity with humans and chimpanzees. Okay, so they are related, and there are some species they are not very related with humans. So likewise, they develop trees, gene trees like this, or phylogenetic trees, looking at this relationship. So that is called cytogenetics. So what are the techniques available to develop this kind of trees from gene related or other data? So that would be discussed in cytogenetics. And protein and RNA structure prediction. So proteins are the main functioning body in humans and all the other organisms. So once we have the protein sequence, so you know about proteins, isn't it? In your advanced level, not advanced level, I think in all levels you learn about proteins. It's a chain of amino acids. So these amino acids, how they are combined with one another, and how they, after they combine, they fold into different patterns like this. So this folding can be predicted using computational tools. So what are the methods available and how these methods are implemented would be discussed under this topic. Okay. And then proteomics and next generation sequencing techniques, how these genes or proteins are sequenced is discussed under this topic, and this available data analysis methods would also be discussed. Then mRNA data analysis and drug discovery, how these drugs are discovered, what are the basic algorithms used, and what are the techniques used would be discussed here. Okay, so very interesting set of topics should be discussed in this lecture series. These are the recommended books. Most of these books are available in the library if you go, and there are some copies are available online as well. So I would recommend you to read these books. And apart from these books, I also give you some articles. Okay. So after today's lecture, I will make around 2 articles available for you to read about bioinformatics and its applications. Okay. Okay, so give me a second. All right. So let's talk about the biology background required. Mainly will be, this course is based on the genetic and proteomic and transcriptomic data available, okay? So you would not have an idea about what we are dealing with. So when you are doing this, if you get some research area, you work with some data, isn't it? As an example, if you get a newspaper article classification process, okay, what do we deal with? You deal with the words, isn't it? Words or sentences or language features, okay? So likewise in this broad bioinformatics area, in this particular subject which you are studying, we'll be focusing mainly on the sequences, gene level, in genetic level, and then in proteomic level, only on the set of genes or gene sequence. What is a gene? What is gene sequence? Actually, you know, genes are built up of certain chemical components. Okay, so they are called nucleic acids. So we'll be discussing about them later. So I just wanted to give you some idea what we are working with on this subject. Okay, so as an example, I will write a sample gene like this. Okay. Like this, only a set of letters actually. These letters correspond to those nucleic acids. So this is a sequence, gene sequence. In proteins also, we have a set of letters. So that's what we are dealing with in this bioinformatics subject. So this is gene sequence. So let's go into details. So cells are building blocks of life. So why do we say so? All the living organisms are built up of cells. There are each and every organism has a set of cells, set of, or maybe there are single unicellular organisms as well, like paramecium and amoeba. They have one cell. You have learned about this paramecium and amoeba in O levels, isn't it? So you can see this amoeba and paramecium in this picture, in the top two pictures. They are unicellular. You can say they only have a single cell. Okay. The others, like humans, are multicellular organisms. They have millions of cells, many cells. Okay. So cells can replicate independently. So what is this replication process? So you know there are very thin people, there are fat people, isn't it? And when you eat more and more, you get fat, isn't it? So that is actually done by this cell replication. And also fat also acts, fat you eat also acts a main role. So these muscles and everything is built up of cells. This cell replication happens when nutrient signal is there. Okay, so that's why when you eat more and more, the nutrient signal is there and the environment is better for replication. One cell, so I'll draw a cell like this, cell would divide into, would grow to a particular size and divide into two daughter cells. So this is called the parent cell. These are daughter cells. Okay, so that is called the cell replication. These cells contain hereditary information. So what is hereditary information? You know, you look like your father or mother, isn't it? Sometimes may not. Some people look like their grandparents also. Okay? And some people look like their uncles and aunties and very different from their parents. Okay? And there are problems come in some families who do not have this genetic knowledge about genetics. They quarrel with each other. This is not our child. How can he be not look like you or me? See, there can be many conflicts. Okay, that's mainly because they don't know about this process. This cell replication process in reproduction happens differently. Okay, sometimes your grandparents' genes may have activated in yourself. Sometimes in your grandparents also there are some genes which were sleeping, which were not active, but activated in yourself. That's why you may look like a different person, or you look like your grandparents, or your one of your uncles. Okay, or you may not look like any of these. Okay, you may look like your grandparents you may have never seen. Okay, so this hereditary information is contained in the cell in this nucleus. In the nucleus, is the storage for genes. Okay, so this is necessary for the regulating cell functions and for transmitting information to the next generation. So you get the information from your parents that you got very nice long eyes. Okay, you got very nice teeth. All information is coming from the previous generation. It's coming from generation to generation. So these in below figure you can see some set of red blood cells. Okay, so there you can see components of a cell. So these are real cells. Okay, seen under microscope. So this is the nucleus, and this is the cell membrane, and this is the cytoplasm. So those are some components of cells. So we are organized from, as I told you, we are organized from a set of chemicals. So these are the chemical compounds, and chemical compounds get together and create a molecule. And then, so these molecules get together and they work in a cell, and cells get together and develop tissues, and tissues get together and develop an organ. And then many organs like maybe a stomach, then the liver, they get together and develop a system. As an example, digestive system. It got several organs in its components. Okay, so in the organism level, we have a set of systems together working together. So that's how we are built up of. So but we do study main techniques available for analysis of these molecules, molecule level. Okay, molecular data in this subject. So these are the genes, and so this is how genes are stored in the nucleus. So this is a cell, okay, the same cell we saw here. Okay, this is a real cell. Sorry, this is the real cell. So this is what we draw. And inside that, there's a nucleus. And in the nucleus, I told you, genes reside. Okay. So this is the cell, and these are the chromosomes. In the chromosomes, you can see genes. Genes are packed together to these particular patterns called chromosomes. Okay. And these genes are transcribed and create these proteins. Translation and transcription processes happen and proteins are developed. So what we are applying tools on these genes and proteins mainly, okay, in this course unit. So we have to look at what are the components build up these genes and these proteins. So DNA, which is the long-term deoxyribonucleic acid, is the basic unit of heredity. It is what coming from your father to yourself, your mother to yourself, okay? So there are different genes for characteristics, like there are genes specific for hair color, genes specific for the color of the eye, genes specific for the color of your skin. So likewise, there are genes specific for particular characteristics of yourself. Okay, so that's why sometimes you may get your mother's color and father's eyes. Okay, it's mainly because genes coming from your mother for the skin is activated, and father's genes for the skin is dormant. They are not active. Only one gene coming from one of the parents is active in yourself. Other one is dormant. Other one is inactive. Okay. So DNA is where the genetic information is stored. So if I repeat the things, so if this is the cell, inside the cell there's nucleus. Inside the nucleus there are chromosomes, and in chromosomes, in these chromosomes, so they are like scissors, okay, like this. In these chromosomes, these genes are packed. Okay, proteins are not inside the nucleus. They are actually derived from genes. Okay, manufactured from genes later. So curly hair and blue eyes are inherited by this. Depending on your gene and little differences in the genes, in yourself and in your friends, because of that, these differences can be seen. Your hair may be curly and your friend's hair may not be curly, okay? So these genes, how does it contain this information? They contain the information as a sequence of nucleotides, as I told you earlier. So what are nucleotides? There are certain molecule types like this type, there are only 4 types, okay? That's not very complex. There are four building blocks of genes. What are they? These names are given here. You can see adenine, we mark it as A, guanine, we mark it as G, and thymine, we mark it as T, and cytosine, we mark it as C. Okay, so those are the main building blocks of genes. And in our programs, we use these short terms A, C, T, G. Okay, so genes are built up of connecting these together, these 4 building blocks together repeatedly. You can see like either repeated or not, like A, G, C, C is repeated here. Maybe G again, T, T, T. So likewise, different lengths and different combinations of these 4 building blocks would build up the genes. Okay. So each of these nucleotides is made up of sugar, phosphate group, and base. Okay, you can see each of these building block A, C, T, G has basic building components. What are they? Sugar, a sugar component, a phosphate group. This is the sugar component you can see in the blue color, and the phosphate group in the this indigo or purple color, and in yellow color you can see the nitrogen base. Okay, and if you look at compare these units, this one with this one, what can you see? These two are similar, isn't it? In each and every component, these two are also similar. That is common in all the building blocks. However, if you look at the nitrogen base, this one, this one, this one, and this one, they are different from one another. Okay, so that is what make the differences in these 4 different nucleotide types. Okay, there are 4 different bases: guanine, adenine, thymine, and cytosine. In those 4 different bases, if you look inside in more detail, what is different is only the nitrogen base. Okay, so that's another thing you can observe here. So the information, information coming from one gene to other depends on this combination or the order of these building blocks. Okay, so these genes may be thousands of nucleotides long, and length and the pattern of or the order of these nucleotides would make these genes. Okay, so genes may be many thousands of nucleotides long. So when we get this set of genes of yourself, it's called a genome. Okay, it contains all the genes. So if you get a single cell of yourself inside the nucleus, if you consider all the genes inside this nucleus, which are packed in these chromosomes, it's called the genome. Okay, it contains all the genes which should be required for the vision and which are responsible for this building of yourself, like your muscle genes, and which are in the stomach lining. So all the genes are contained in each and every cell of yourself. So how can that be happen? So you can see, if you look at your nails, its surface is different, isn't it? If you look at your skin, skin is different from your nail. However, I say that all the cells in your body, in your fingers, in your eyes, in your mouth, everywhere, they contain the same set of cells, or not cells actually, same set of genes. Okay. So what would make the differences? If you get a finger, you know the appearance of your finger in different places is different. What would make the difference? If I say that each and every cell, even a cell in here or here or here, these cells would have the same set of genes. If you get the two cells, the set of genes which it contains would be same. So how can cell differ from your hand to your eye? I mean, they have different functions, isn't it? The eye is responsible for the viewing and hand is responsible for other tasks. So how can it be different? This gene expression, the set of genes expressed in skin cell is different from the set of genes expressed or active in an eye. Okay, so that is what makes the difference. However, in each and every cell in your body, they all have same set of genes. Okay, they have all the genes and same set. However, in particular cells, only a set of genes are expressed, and that expression makes that cell specific. Okay, that expression means making a protein process. So proteins which are active in eye cell is different from the proteins which are active in skin cell. Okay, so to further understand this arrangement, okay, if we get a book with several volumes, so what are volumes in a book? What's a volume? Volume, so if I get sharing the whiteboard, so this is your book, so you are writing a new book, and it's very lengthy book. This is part 1, this is part 2. Okay, we call it does have 2 volumes. And if you open up one book, what do we have? We have sentences. In the sentences, what do we have? Letters or words, isn't it? And these words build up sentences. These sentences together build up paragraphs. These paragraphs together build up 1 volume, and 2 volumes build up the book. Okay. So if you try to analyze our genes also with this, okay, if you try to develop analogy, so letters would correspond to nucleotides. Okay, these nucleotides, you can see here, correspond to the letters in your book. Okay, and the sentences are just like genes. This is one gene. There would be other genes. So they would look like sentences in your book, these genes. Okay. Then individual volume of encyclopedia is like chromosomes. These genes packed together into components like this called chromosomes. Okay. These chromosomes look like individual volumes. These volumes get together and build up the book. Okay. These volumes get together and pack in a nucleus and it builds up the genome. Okay, all the volumes, all the nucleotides with all the genes in your cells build up this genome. Okay, so what's genome? Genome is which contains all the genes required for an organism, not only for humans, maybe for other organisms. It contains all the genes. Okay, so that is the analogy. Okay, so these are chromosomes. So these are packed in the nucleus of the cell. Okay, so these are human chromosomes. These chromosomes are made up of DNA strings. So you know these genes are packed together like spirals. You know spiral, isn't it? This is a spiral. These genes are packed together, one after the other, and build up these chromosomes. So each contains genes in linear order, one after the other, like this. Okay. So this is one gene. This is the second one. Likewise. So human body contains 46 chromosomes in 23 pairs. There are pairs. If you count, there would be 23 pairs. You can see there are 22, and with the sex chromosome, either XX or XY, XX would make a female and XY would make a male. It contains 23 pairs. Okay. So chromosome pairs 1 to 22 are called autosomes. And this one is called sex chromosomes. Okay. And there are certain chromosomes which would carry genes corresponding to particular gene. Which should carry genes corresponding to particular disease. So this is the chromosome. In chromosome 11, you can find the gene which corresponds to sickle cell disease. Okay, and now there are new technologies developed that you can cut the particular gene from your genome. Okay, there are studies likewise, and one of your seniors did the research on that gene cutting process. Okay, last year. Okay, so this is how it is. I mean, again, if you look at the basic level, these are the basic building blocks: adenine, thymine, and how they are packed like a spiral. So as I told you, there's this gene, that these genes are packed in a spiral. See? So from here to here may be one gene. Here to here may be second gene. So likewise, these are packed. So if you go into more detail, so if you zoom this section, you can see something like this. So these colors correspond to these amino acids or nucleotides, adenine, thymine, likewise. Not amino acids, sorry, nucleic acids. Okay. Adenine, thymine shown in purple color, adenine shown in green color. So yellow color, these colors are different from here to here. However, these four colors correspond to the 4 nucleic acids. So you can see although we write a single set of letters like A, T, G, C, C, like that in your gene, always there's another strand as well. So this is only a single strand. So you can see this one, this section, this get into spiral shape and makes this, okay? So if you look at, divide this into two. So there are adenine and thymine. Adenine is coupled with thymine always. That's a rule, okay? Adenine with thymine and guanine with cytosine. So this is guanine and this is cytosine, you can see. And this is again thymine and this is adenine. Okay, so this is guanine and cytosine. So these are coupled together likewise. Although we write only one strand as a gene. As a gene, what we write is a single strand. But if you look at the real structure in your body, there's another strand as well coupled together, bundled together, and forms a spiral because of that. Okay. So if you look at this, it would contain two strands rather than one. So as an example, if we get AATGC is one strand, and in the other side it should have, from in A, it should couple with T, A should couple with T should couple with A, G should couple with C, and C should couple with G. So if you are given one strand, it's possible to write the other strand, if you know this. So you have to know this. Okay, guanine is always coupled with cytosine and adenine is always coupled with thymine. Okay, this DNA double helix has a phosphoryl end called 5N and hydroxyl end called 3N. Okay, this is the 5N, this is the 3N. Always in nucleic acid, you can see those 3 ends. So when you are writing the that complementary strand or the other strand of a given gene, always it's opposite. Okay, if you look at this, 3 and 5 end, 5 and 3 end. Okay, A with T, A with T, T with A, G with C, G with C, and C with G. So if you are given something like this, T, G, C, C, A, sorry, A with 5 end and 3 end marks like this, you should be able to write the complementary strand of this. So you can write to 5 end, you should have 3 end. So what you are writing actually this one. We are given this one with 5 end and 3 end. What you are writing is this one. So we have 3 end and 5 end at the opposite. T would bind with A and G would bind with C, C would bind with G, again C would bind with G, and A would bind with T. And 5 end is on the other side. Okay. So should be able to do this. If you are given something and if you know this, you can do that. Okay. Okay, now let's look at, now you have some idea about DNA, isn't it? There are 4 main building blocks and these building blocks get together and build up genes with different lengths, with different order. Okay, so that's it. That's the building block. So what are proteins? The other main component we will be studying is proteins. What are proteins and what are proteins built up of? these proteins also have building blocks. So, you know, in genes, we have 4 building blocks now. 1, 2, 3, 4. So in genes, no, in protein, it's not like that. In genes, we have only 4. In proteins, we have now 21 if you search in Wikipedia, I think. When we were studying, it was 20. But if you look at this new research, they have found out another very another basic amino acid. However, I will consider as 20 in this course, okay? Okay. Proteins also are given different names like glycines, alanine, valine, likewise. Okay, so if you look at proteins, also genes contain hereditary information. Protein are the functioning body. Okay, so that's what I previously also tried to convince you. So in skin cell and in eye cell, we have cells with the same set of genes, isn't it? So how does these two cells differ? It differs from the genes which are active in these two cells. Okay, so this activity means expression. Expression means genes contain the information. That information is converted into proteins. That conversion process is called gene expression. Okay, so we'll be discussing about gene expression further later. So after this gene expression, the final product is the protein. Okay, this information contained in a gene is not active as far as there's no any protein created from that information. Okay, so the function of a protein depend on the order of the function of the protein would depend on particular gene expressed. Okay, different genes express different proteins. So if this is a gene, and in the expression process, proteins are created. Okay, so these proteins would depend on the order of the amino acids. So these proteins are built up of amino acids, this 20. So if you look at the variability of proteins is very much higher than the variability of what? DNA. The variability of proteins, if you see there are 20 basic proteins available, they would arrange in different orders. Okay. So, and since there are 20, there are different number of, there are very large number of combinations than you have for genes. Okay. So the variability of proteins in your body is very much higher than the variability of genes. Okay. So the information required to make amino acids stored in a DNA. So a DNA sequence would determine amino acid sequence. So this, let's look at that, how a DNA sequence would determine amino acid sequence. Okay. Then this amino acid sequence would determine the protein structure. Then protein structure would determine protein function. So that's how this information in the gene is converted into a protein, and then these proteins are the functional body of the cells which are responsible for the particular functions, giving you the eye color and giving you the vision. Not vision actually, vision is a collective process. So skin color, likewise, curly hair, likewise. So when the conversion happens, this is a gene and this is a protein. Protein also build up of these small building blocks called amino acids. So this conversion process, in the conversion process, there's intermediate product produced called RNA. The storage is done in DNA, and DNA would not be destroyed. A copy of the DNA would be created, and then that copy is transferred into, it is the information transferring component, which is the RNA. And then using that, proteins would be created. Okay, so that's the process. So this is the central, this is called central dogma, which is the gene expression actually. Another word which is commonly used in biology is central dogma. Okay, in molecular biology. This is central thing in our body. Okay, you should know actually. So these are the genes. This is a copy of the gene created. And from this gene, it gets together with a component called ribosome and creates proteins. Okay. So this is a gene and this is the copy created, and that copy comes out of the nucleus and then get together with ribosome and creates a set of amino acids bound together, which would make a protein at the end. And it folds in different patterns, and after it folds into its most comfortable shape, it would be responsible for a particular function of your body. Okay, so if you write this in component wise, DNA would go through process called transcription. In the transcription, RNA is created, which is a copy of the information contained in a gene. Again, it is you can see set of basic building blocks of nucleic acids, and then translation happens and proteins are created. Okay, so if you get the DNA, what is DNA actually? It is a set of nucleic acids, 4 nucleic acids, T, A, C, G. They are arranged together in different lengths. So this is only a component or part of the DNA. So that part is converted or copied. So how this is copied? This is the complementary strand. Okay, T with A with U, C with G likewise. Okay, now you can't see T in RNA. You can see instead U. Okay, if you look at this, G would replace with C, A would replace with T. So this is the copy is actually a complementary strand of this, like this. Okay, you can see that T will replace with A, T replace with A will replace with U. Okay, likewise. So after that, we get 3 nucleic acids at once, and that would create 1 amino acid, not create actually, that would correspond to 1 amino acid. This corresponds to 1 amino acid, likewise. So this correspondence can be seen in a table, like, this table. Okay. This is the first letter found in the, not letter, actual building blocks found in the RNA. This is the RNA. So in RNA, this is the first one, A, U, G is the first codon. We call this 3 units a codon. So that codon, the first letter is A, isn't it? That is that, AUG. First is A, second is U, third is G. A, U, and G. Okay, which should correspond to mate. Okay, so likewise, each codon would correspond to a particular amino acid. So met is a amino acid, one of the 20 amino acids. So what is met? This one. This amino acid. Okay, that is built up of what? AUG, which corresponds to TAC. In your gene, in the expression process, if you have TAC, it would create MET, nucleic acid. Okay, this amino acid. This amino acid get together and build up the protein. Then this should fold into particular pattern and correspond to a particular function in your body. Okay, DNA makes RNA and RNA makes proteins like that. Any problems? You can now ask me. If this process of conversion is not clear, please let me know. And you are given a time to discuss with each other in your group activity as well. So there also you can talk with your friend and clear up the things. And it's better to clear up the things now because I'll try to help you. Any problems? Oh, all the things clear for you? So up to now, what we have discussed is actually this. DNA is built up of what? Nucleic acids. How many basic components are there? There are 4 basic components, A, C, T, and G. So these are actually these genes are built up like arrangement in different order and different length of these 4 nucleic acids. Okay. So after that, we looked at proteins. Proteins are built up like this. DNA is converted into RNA, and then RNA is converted to a protein like this. So that process also very transparent. There are no hidden things happening. There are some complex things happening. However, basically this is the basic expression which happens. Okay, so if this is the DNA, this is the complementary strand. T is replaced with A, not replaced. A copy is created. If we have T here in the copy, it would create A. So this is the copy now. So in this copy, it is a complementary strand. So you can see here. This would look like more like this. So this is the complementary strand of this strand. T, A, U, C, G likewise. And then if you look at this chart, you can identify each and every amino acid which correspond to these codons. So there are 20 amino acids. So one nucleotide cannot correspond to one amino acid. Why? Because there are 20 amino acids. We have only 4 building blocks. Okay. It cannot correspond to 2 as well. Okay. So if you look at the combinations, you can write A, C, A, T, likewise. How many combinations can you write with 2? 2 amino acids. It is less than 20 if you write. Okay, so that's why we can't get two groups. However, we can go to 3 groups, so triplet codons. So that's why we go for triplet codons. When there are 3, there are combinations we can write which is more than 20. Okay, so because of that reason, so you can see how nature is working, isn't it? It's amazing. So these three sets would correspond to one amino acid because of that reason. Okay, these codons that do not correspond to a protein are called stop codons. Some of these three groups, so you know when you make combinations, there are more than 20. We can make with 4 different letters, A, C, T, G. And these 4 different letters, when you arrange into 3 by 3, A, C, T likewise, you can go for different combinations with more than 20. So these 20 of these correspond to amino acids, and more than 20, the other codons we use for different other functions, like stop codons. These stop codons are places where genes start and stop. You know, genes are arranged one after the other, and we don't know where a gene start and where a gene stop. Okay, so for that also, there are particular codons or three amino acid groups available to identify that stopping points and starting points. Okay. So starting points are called start codons. Stopping of the genes is called stop codons. Okay. Okay. So now you should have some idea of gene expression. A single nucleotide cannot be taken to point out the beginning of a gene. So a particular combination is put out. So promoter sequence is the order of nucleotides which mark the beginning of a gene. So as I told you, so these 3 codons, although it points the start, not only 3, there's a region, okay, which is called the promoter region. So these genes are not always activated. So see, only in some cells these genes are activated. If this particular section is activated, only some component would come and bind into this section, and then only this would start this transcription process, translation process, and hence this creation of proteins. Okay. So this gene expression is the process of transcripting a DNA and translating a RNA to make a protein. Simply this process, what we have discussed here. Any problems? We are going to look at a nice video. Are you there or are you sleeping? May we go for the video? Mahoga, any problems?
Madam, I have one problem.
Yes.
A always convert to U.
Yeah.
Always.
No, not always. A is not convert actually. There it is created. So if you look at this, okay, I'll try to explain this in this way. If you look at the DNA double helix, okay, so this is called double helix because you can see one of this is a helix. Okay, 2 of the helixes are get together and forms a spiral shape. So if you look at this DNA, the real DNA, okay, in the DNA, A is combined with T always. In the other side, if you see A, which is in green color, always you can see the yellow color on the other side, T, isn't it? So that is in the DNA. If you look at the RNA, always A would create it either with U here, and if you have T in the original, it would contain with, it would create A here. It's the copy actually. Okay, it changes, this U changes, replacing T only in RNA. Okay, don't be confused. In DNA, it is always this combination. G with C, A with T. Okay, G with C, A with T. However, here in RNA, always when you have A, it would create this copy as U. Okay, complementary one, no. The complementary one, no, is not T, but it is U. Okay, so this is a copy created, but copy is the complementary one, not the original one. It is the coupled one. Okay, coupled one. T with A, G with C. Like us, A is not with, if you have A in the DNA, you would have U in the RNA. Okay, but in a gene, if you are asked to write the complementary strand, should be T. Clear?
Yes, madam. Thank you.
Okay. Others? Any problems? Otherwise, we can go for the group work. So they are also, you would you would have time to discuss with your friends, okay?
Madam, hello.
Yes.
Madam, can we have the recording?
What?
Madam, can we have the recording? I left the meeting several times.
Okay,
I
okay.
can't.
I will let it available for you.
Okay, thank you.
So I'm trying to play the video for you, okay?
Okay.
Now, if there are no any other problems. Voice are very silent, isn't it? There are very nice videos of this gene expression available in YouTube, okay? So you can have a look at that. You look in YouTube for Can you see?
Yes, ma'am.
Yes, sir.
Okay. So tell me whether you can hear also. Can you hear? Did you hear something? Did you hear something? No? Mahoga, did you hear something?
No, madam, I think.
I don't know why.
Sorry, madam, we play one.
But pardon? Mahoga, what did you say?
Can you madam play
Play
more?
once again.
Can you hear now?
Yeah, we can. Yeah, I think.
Can you hear, Mahoga?
Now we can.
You can't.
Can.
You can, okay.
Madam, can't now.
Yeah, you can't hear, but I don't know why. So I will explain, okay? So these are cells actually. So you know cells get different signals, like as I told you previously. So some cells, as previously also I told you, that when you get fat, when there are nutrient signals, when you eat more and more, these cells get the signals that they can divide. They can divide. So likewise, there are signals coming. So you can see these signals are also coming as particular molecular components. So now you can see inside the cell. So this is what the this is how the outer signal came. Okay, now in the cell also there are components which will take the signals from outside. This white color is something like that. That particular component would take the signal into the nucleus. So this is the nucleus. So that would go into the nucleus, and you can see now these are the genes, okay? And that's white color component is attached to the promoter region. Promoter region is the region which is responsible for the activation of the gene. If the gene is not activated, it would not express and create proteins, okay? And so this is the process of creating a copy or the RNA. So as a RNA, what we get is a complementary strand of this particular DNA, okay? So now a copy is only taken outside the, okay, now, and this RNA is also, although I didn't tell you previously, some components are removed. This is the RNA, and you can see this blue color section is removed. Now it's taken out of the nucleus, and then the translation happens, and these are the proteins created. Now that proteins can come outside the cell and create necessary activities that can come outside also, that can reside inside. Okay. So I have actually this in LMS. So now you have access to the LMS, isn't it? Yesterday they activated it. Did you try?
Yes, sir.
Okay, so I will put the video link into the LMS. I think it's already there, but it's hidden from you. So, so you can have a look at that, and you can hear the sound as well. And now there's a group activity, but the time is not enough, I think. So do you have a lecture in the afternoon? Anshavis told me that she's going to take the afternoon. So I think we had to stop at 12, maybe 12:15. Would that be okay? Or else I'll give this as a homework. So then you can prepare presentations for next week. It will be better, isn't it? So there are how many people? 7 people are there. So I'll make groups like this. Who is Madhushani? Madhushani, Mevan, Ashfa, Dilan, Hashini, Mahogas, Shan. So I think I would like to give you the opportunity to build up groups of 2 or 3 people each. So you can't go into 2 because there are 7 people. In one group, there should be 3 people. Okay. That group should do the presentation about gene translation. Okay. Gene translation. So the group who have 3 people, I'm not going to name the people. Okay. So you can get together and discuss who is going to be in your groups. Then apart from that three student group, you should have 2 other groups. The other 2 groups should talk about gene transcription. Okay. Make a nice presentation like what we have seen. Don't take much time, maybe a 10 minutes presentation each for 2 people's groups and 3 people groups, 15 minutes. Altogether, it's going to be 30 minutes presentation time for all. Okay. One person can talk around 5 minutes. Okay. 2 people groups should do gene transcription, and gene translation 3 people, because gene translation there are, I mean, you have, it's very broad. I mean, creation of proteins, you know, the proteins are different as well. The variation is very different in proteins, as well as there are different mechanisms happening also. So if you want to broaden up, you can. That's why I give it to 3 people group. Okay. So I think we will continue the lecture until 12 o'clock, and next week you can do the presentations. Okay, so this is the group activity. I think you can see the screen, can't you? Get into groups of 2 or 3, select one of the topics below and do a presentation of 10 minutes. So the 3 people group would have 15 minutes, 5 minutes each for each person. Okay, so so there are groups like this, 2 people, 2 people, 3 people, no, altogether there are 7 people. So these 2 groups are doing about gene transcription separately. Okay, they would talk about gene transcription from start to end, and again group 2 will repeat about the same topic, different presentation style and content. Okay, any problems? Anything not clear for you? If there are any unclear?
Yes, Madam Clear.
Okay. So now we'll be looking at some applications of bioinformatics to further motivate you. So these are the common activities done in bioinformatics: mapping and analyzing DNA and protein sequences. So what is this process? So this mapping process, it would actually try to identify where the DNA starts, where the DNA stops, where protein starts and where protein ends, and what are the functional regions of protein, what is actually responsible for the function of the protein. If you identify that, you can if some person have a disease related with the particular protein, we can remove this. I mean, you can do some we can apply some drug on this particular region. So likewise, we can do things to avoid that. So that is one of the common activity we do, mapping and analyzing DNA. So likewise, we are trying to identify promoter regions, what were the gene ends, what are the regions which would code these active regions of protein. So likewise, mapping process and analyzing process would happen. And align DNA and protein sequence to compare differences and similarities. Like I would like to compare my part of gene with my father's some components to check whether I also would have the disease as my father has. Okay, assume that one of your, maybe your long relation may have some genetic disease. Okay, and you are also interested whether you also have that disease, like cancer, sometimes it comes genetically. So once you want to do that, you need to compare your particular gene corresponding to that cancer with the particular gene of your relative. Okay, so that is done by comparing. We just keep the first DNA here and the second DNA in the next row, and then compare the components. Okay, and if there are similar regions, it means you may also have the particular disease. Okay, in future. So aligning DNA and protein sequences is a process something like that. And creating and viewing 3D models of protein structures. So protein is a set of what? Amino acids. There are 20 amino acids. So once they arrange one after the other in particular order, in particular length, it would create a sequence of amino acid, which is called the primary structure of the protein. Okay, so this is the primary structure, and they would fold into different patterns. This primary structure is folded into different patterns. This although human they are people, you sit down in a different way than your friend. You sit down in the way you prefer and where you feel, when you feel the most comfortable way. Okay, you would sit down in the way you want and the way you would feel the most comfortability. Okay, so these proteins are also like that. They have this long strand of amino acids, and they would fold in a particular pattern depending on the bonds in between these chemical components. There are bonds in between the chemical components, and this is the most comfortable way, most comfortable and lowest energy state, in physics and in normal day-to-day life also. If you throw a ball into a playground, where would it stop? It will stop at the lowest energy position, isn't it? If you throw a ball, it would fall and roll and stop at the lowest energy position if there are no other obstacles. Okay, it depends on the environment as well. So here also it would fold. This protein would fold and remain in its most comfortable, lowest energy state. So predicting this can be done by computational algorithms. Okay, we can't do it actually by manual process. You can't do this. You can only observe that under the microscope. So to do this, people have written algorithms. They are not very accurate at the moment also. Research is going on. And if you look at the web, there are online competitions which would give you a lot of money. If you can predict the folding pattern of particular, there are coding competitions available for people to join and predict the folding pattern of a particular protein provided for each year. Okay, you can also have a look at it. So this is a very current research field in the bioinformatics field. Okay, so creating 3D models of proteins. Oh, there are, we are looking at bioinformatics techniques. So in database also, database related bioinformatics tasks are also there. So you know the sequences are stored in databases. So we'll be looking at those databases next week. These databases are free to use. Okay, so there are some very rich countries like USA, UK, and Japan. They maintain these databases with DNA information, so that you all can retrieve this information freely. Okay, so that's a great advantage. Why do they? America, these UK people don't do the crazy things. They would not just give you information as, thinking about us or the world. Okay, so what they do is actually this huge amount of information cannot be processed by themselves only. Okay, so this is a collective process. So that's why they make this information freely available for you. So it is, anywhere in the world, if a new DNA sequence is discovered, they give the opportunity for them to, anyone in the world, to upload that sequence. These biological people who are working in the lab can upload freely to those databases. So the people can, like bioinformaticians, the other people who are interested can download and do research. Okay, so there are many databases like that. So these databases are located at different places. As I told you, in America, mainly in UK, like rich countries. And they exchange information on a daily basis so that they are up to date. So this database they also exchange information. If they have a new sequence, they give to the other databases as well. So these primary databases collect sequence data like DNA sequences, protein sequences, and the derived data like cancer-related data are secondary databases. Okay, so in primary databases, you can find sequence data, especially of DNA and protein. Okay, so we have now sequenced and identified genes. So we know what they do as well. So these sequences are stored in these databases. So if we find a new gene in the human genome, we should be able to compare it with the available genes. Okay. So now from the Human Genome Project, so those are the keywords. So you need to search in the web what is Human Genome Project. So it was initiated a long time ago and it was completed. So that the main target of this Human Genome Project was collecting the whole genes, the whole set of genes of these humans. Okay. So they have collected to some extent and they say they have finished. Still they are doing the updates if they find new genes. So what would happen if you find a new gene as a biologist? Assume that you are a biologist and you found a new gene. Okay. So you may need to check whether that is already available in the database, isn't it? So what you have to do is you have to compare each and every gene in the database with your new one. Okay. So that is called database searching. For different purposes we search. This is one of the main purposes we do these database searches. Since there are a very large number of databases, we cannot do sequence alignment for each and every sequence. So sequence alignments one by one, it would be difficult. Okay. Comparing this one with A. Likewise, okay, it's quite difficult with a large number of sequences like this. So there are heuristic algorithms we need to develop to these purposes. There are algorithms available. We'll be talking about sequence alignment algorithms later. Still, there's a need for heuristic algorithms because that takes a very, very lot of strong data. Like in a computer, in a normal computer, we can't do such thing without having a heuristic algorithm. It means we can't do one alignment with the other one as we do manually. Okay, we should have shortcuts. We should use some special techniques. So based on those techniques, we should write new algorithms. Okay. Okay, still we have another 10 minutes. Okay, so let's look at some areas in bioinformatics research. It's better if you do this after the presentations. You should have a good idea about this gene expression process. So I will postpone it to next week. So after you do the presentations, we can continue. Okay. I think I would stop the lecture from now and I'll make these lecture notes and also reading materials available in the LMS. Okay. And also the recording of today's lesson. Okay, any problems? If you have any problems, you can ask me now. I mean, without talking, we can't do this. You know, it's not like a physical class. I can't see your faces because I don't have any idea about if you don't tell me about your understanding. Any problems? Any problems about the group work? Do I need to name the people? I feel very boring when you don't talk. Okay, I'll stop the share and end the meeting. And thank you very much for participating for the lecture today. And have a nice day. And if you have any problems, you can email me. If you are very shy to talk, you can even send to the chat. But I